All right, I think we're good to start. So, good afternoon if you're joining us from Australia. Good morning if you're in Israel right now. And thank you for joining our third special edition coronavirus live broadcast. My name is Ori Danieli. I'm the Executive Director of Technion Australia. Those of you who are not familiar with Technion, it's Israel's Institute of Technology and among the world's leading dedicated to science, technology, and engineering. Before we start, I wanted on behalf of Technion Australia, Technion in Israel, our event partners, North Sydney Innovation Network, to firstly share our deepest sympathy to anyone who has been affected by the coronavirus in any shape, form, or any other way. And in particular, if you lost someone close, of course, or if you or anyone close to you is unwell, we wish you a very speedy recovery. Before I introduce our distinguished speakers, I would like to welcome our partners for this webinar, North Sydney Innovation Network, and to ask Jono Herman, Chairman and Founder, to say a few words. Jono, over to you.
Thanks, Ori, and thank you everyone attending. So North Sydney Innovation Network aims to educate, network and advocate for startups and technical businesses, innovation-rich businesses on the North Shore of Sydney. We're very proud to bring such expertise through Technion to those who will attend here or watch us on Facebook in our group. And we thank Ori for that opportunity. So back to you, Ori.
Thank you, Jono. So there's one thing that's spreading faster than the virus, and that's the search for a vaccine or other health-related aspects that can reduce the spread of the disease or increase the way or improve the way that we test or become more efficient with this epidemic. So we're very fortunate today to host three amazing experts, scientists, who are very much involved with research related to COVID-19. And we know that you're very, very busy, so we really appreciate you giving up your time this afternoon. We've got Professor Alon, who is the Vice President of the university, and he'll talk about some research that's going on at the Technion and life at the academia and life in Israel in general at the current state. Professor Michael Wallach, who many of you have those who have joined our previous webinars would have met. Michael is a professor at UTS. He's also a visiting professor at the Technion and a global expert on infectious diseases. And we've got Professor Debbie Lindell, who is from the Faculty of Biology at the Technion. Debbie is originally from Melbourne, and she'll share with us a little bit more about the science and biology of the virus, including explaining perhaps how the coronavirus compares to previous global medical outbreaks such as SARS and Ebola and others, and talk a little bit about the challenges associated with this particular virus. So the way today is going to work in general is each speaker will speak for approximately 10 minutes, and then we'll open it up to the crowd to ask questions. So if you have questions, you can post them straight away. You don't have to wait till the end, but we will allocate the time for following their presentations. So our first speaker today is Debbie Lindell. Debbie, thank you for joining us. At our last webinar, which was only over a week ago, I read the numbers of confirmed cases, which was around 422,000 globally and close to 19,000 deaths. We're now at just over 1.2 million confirmed cases and over close to 68,000 deaths, meaning we pretty much tripled in the last 10 days, those numbers. So I'd like to kind of throw that at you as a starting point, if you can reflect on that and then go into your presentation, please. Thank you, Debbie.
Okay, thank you very much, Ori, and good afternoon to everybody there in Australia. Or you want to share the screen?
Absolutely, yeah.
Terrific. So as everybody is very well aware, this pandemic has really affected the entire world, and there's over 200 countries that have been affected. You're in presentation?
Yeah.
Okay, so over 200 countries have been affected, and this pandemic began in China with an outbreak that led to over 80,000 active cases at one particular time. But then the outbreak spread to Europe, as you can see here in the yellow, and then to the United States, and it spread in a very alarming way. But we can take we can take a bit of comfort from what happened in China, in that China instigated directives that led to bringing the outbreak under control. And in reality, both Israel and Australia took the lead from China. And even though it started out both in Israel and in Australia quite rapidly, increasing exponentially, both countries put into place social distancing and lockdown that has really led to bringing the case number under control. We still need to, at least in Israel, see it decline, but I personally am optimistic that we're in the right direction here. Okay, that's not working. It's not, can you, maybe I'll have to do.
I'll move to the next slide.
Yeah, if you can do that.
Yeah, is this the one?
I've just did.
Try now, try now.
Okay. No, it's not moving.
All right, are we on the right screen now?
It's not moving.
I can move it for you. Just tell me which slide.
Maybe that's best. Maybe just go ahead and move it now.
All right.
Okay.
Is this the one?
You have the
next one.
I don't have any change. Other people got change in the slide?
Jono?
Maybe I should just share my screen.
Yeah, try that. Is it sharing?
Yeah, I'll do it in a sec.
Sorry, guys, bit of technical error. All right, it's all good.
Yes. You see my, yeah. Okay. Okay.
Perfect.
So I want to actually start by describing a little bit about the biology of viruses in general, and this particular virus, to understand why we have problems really in finding ways to combat it, and why it takes some time to deal with new viruses as they come into populations. So first of all, a virus is basically a virus is basically a genome which encodes the instructions for virus replication. And this genome is protected by a protein coat that protects this genome in the environment. And then in the case of many viruses, including the coronavirus family, there is another envelope, a lipid or fatty envelope surrounding this, the genome and the protein coat, that contains proteins in it that are used to recognize and enter into the host cell. Now, viruses are obligatory parasites. They really can't reproduce outside of a living cell. So they have to be able to enter into a cell, and then once they enter into the cell, they hijack the cell's own systems to be able to replicate. And they use this to turn the cell into a factory to produce new viruses. Now, the infection cycle begins by the virus recognizing a protein on the cell surface. Now, these proteins on our cell surfaces aren't there for viruses. They're there for our own needs, and they play essential parts. Viruses take advantage of this and use them to enter into the cell. And then once they've entered into the cell, they replicate, they make the proteins needed to replicate their genome. They replicate their genome, then they assemble the genome and the proteins around it inside the cell. They take on a membrane, on a lipid envelope, and exit from the cell and kill the cell as they do so. So there are 7 known coronaviruses that infect humans, 4 of which cause the common cold and are not really serious in most cases. And there's been 3 others that have caused more problems, including the one that causes COVID-19, and that's the SARS-CoV-2, the last one in the list. So one of the things that scientists wanted to know, and in fact the entire population wanted to know, is where did this virus come from that it entered into our population? And the way that this is done is to look at their genomes and to compare their genomes to other human viruses and other coronaviruses that infect animals, which they're known to do. And what was found by doing a comparison across the entire genome was that the viruses that infect the humans, these here in red, are most similar to those of a bat virus throughout the entire genome, and quite different to the other human viruses that causes SARS, MERS, or the others that cause the common cold. So this has provided information that this virus likely originated from bats, but there's also some evidence to suggest that certain parts of the genome have come from a virus that also infects pangolins and has been swapped in and out. Now, what has enabled this particular virus to infect humans is likely changes that have occurred to the protein that is used by the virus to recognize our cellular proteins. That's allowed it to recognize human cells and infect them. And so what's happened is we've got a new virus that's entered into what we call a naive human population that has no natural immunity because we've never met this virus before. And nor do we have vaccines against this virus because we've never had to develop one because we've never had to deal with it. And so this has actually allowed this virus to spread quite easily throughout the human population globally. So if we now compare the virus that causes COVID-19 here on the left to viruses that we're a little bit more familiar with, for example, the seasonal flu virus. And we look at 2 parameters that are often used in epidemiology to describe the severity or the likely problematic nature of an emerging disease. One is the transmission number. How many people are affected or infected by any particular infected person on average? And the mortality rate, the case fatality rate of those that have been infected. And if we look at influenza, which is something that we're quite familiar with, the seasonal influenza flu is transmitted at a rate of approximately 1.3 to 1.5. Above 1 means that it could cause an epidemic. And the mortality rate is about 0.1. If we compare these values to the COVID-19, we see that transmission is much more rapid, 2 to 3 times the level. Mortality is about 30-fold higher. So we do need to take this virus seriously in comparison to the seasonal flu. If we compare it to Ebola, though, we can see that Ebola is much, much, much more dangerous if you're infected. Nearly 1 in every 2 people will die of Ebola if they're infected, whereas we're not talking anywhere near those levels with COVID-19. On the other hand, transmission is through droplets in the air in COVID-19, so its transmission is very good, whereas Ebola, you need person-to-person contact. But I want to actually spend the last little bit comparing it to this SARS virus that came out in 2003, which is the same family of viruses, and try to understand why COVID-19 is causing more problems than SARS. So SARS had a transmission rate of 2 to 4, which is very similar to what we have for COVID-19. And its mortality rate is actually higher, 2 2 to 3-fold higher. Yet SARS killed only 800 people and infected only 8,000 versus the over 1 million people and 70,000 people it has killed so far. So what's the difference between SARS and COVID-19? Why did we manage to contain SARS so easily, but COVID-19 we haven't been able to? And this mainly comes from the fact that people are infectious with SARS when they're showing symptoms. So once somebody showed symptoms, we could take that person and isolate them and prevent them from infecting other people. In comparison, COVID-19 is infectious prior to somebody showing symptoms, which means that somebody can be sick and they don't know it, and they can be transmitting the disease, and therefore it's much more difficult to prevent transmission. And in fact, even people who don't show symptoms can transmit the disease. So this is actually the reason that social distancing on a large scale and lockdown is necessary, because we can be infectious and contagious and not even know it and transfer it to other people. So what are we trying to do beyond social distancing to try and deal with this? First of all, social distancing is used to try to reduce transmission to levels below one person infecting more than one other person, so that we can prevent it from spreading further throughout any country or the world. So there's two main ways beyond social distancing. One is finding effective medicines, and the other is developing a vaccine. Now, developing medicines is very difficult. First thing that everybody went to is using existing antivirals, but this is often difficult because viruses aren't all the same. Each family of viruses do things a little bit differently. They all take over our cells. They all use our cells to replicate, but each family does it a little differently. So an existing antiviral against one family of viruses or one type of virus won't necessarily work against the current family of viruses or the current COVID-19 virus, which is actually what seems to be the case. So we need to develop new medicines. And why is this also difficult in general to find medicines against viruses? It's because viruses use our cells. They use our particular processes that we need for our own cellular health and our own bodily health. So if we target them, we're going to be targeting things that we need, and then they can be very problematic and dangerous to us. So we have to go in and target processes that are specific for viruses. So, and these viruses are different. So each time we have to target a specific process that is specific to a particular virus. And we can't use antibiotics because viruses aren't bacteria. Bacteria grow and they make their own proteins, whereas viruses don't grow. They make their parts and assemble them, and they use the cell's protein processes. So antibiotics that target those two processes in bacteria can't be used against viruses. So of course, the holy grail really is to develop vaccines. And I'm not really going to talk about that at all because Michael will be talking about that in a little while. I just want to say that one of the proteins that this family of viruses have is good news for making vaccines. This particular family of viruses has a protein that reduces the mistakes made during replication. In other words, it reduces mutations, so they don't change nearly as rapidly as the flu virus does. So once a vaccine is found, chances are very high that this vaccine will work for many years against this virus, and we won't have to renew and remake these vaccines on a yearly basis. And I just want to end by saying that if science has brought HIV under control, which is a very, very serious virus, I am convinced that it's just a matter of time of getting COVID-19 under control. And in the meantime, us listening to directives and behaving in ways that can reduce transmission. And that's really all I have to say at this point in time.
Excellent. Thank you so much, Debbie.